Respiration in living cells is an exothermic reaction. That means it's a reaction that releases energy, and that energy is useful because it can be used in other reactions that require energy. The energy is released from a molecule called glucose. So where does glucose come from so that animals can release energy from the glucose in respiration? Well, animals consume glucose and then they release the energy from glucose in respiration. Plants also need energy, so that means that they need to use glucose to release that energy. So where do they get the glucose from? They don't consume glucose, so instead they have to produce glucose. When they produce glucose, they can then use the glucose in respiration to release energy. However, they could also convert glucose into other substances that the plant needs. Glucose is a monomer, so you could join the glucose units together to make a polymer, such as starch, which is the polymer that glucose is stored as in plants, or cellulose, which forms the cell walls. Glucose can also be converted to sucrose, which is transported around the plant. It can also be converted to other molecules such as proteins and lipids. Effectively, plants can use glucose not only in respiration to release energy, but they could also use the glucose to make their own material. So the material in an organism is known as biomass, and plants and algae produce their own biomass, and that is the biomass that we consume. So when we eat plants, we're consuming the biomass that they produce from glucose. So plants are known as producers in the food chain because they produce their own food and they also produce their own biomass that we consume. So where does the reactants come from to produce glucose in photosynthesis? We know that the reactants don't come from the soil, as shown by this famous experiment from Van Helmont. Instead, they come from the air, which is carbon dioxide, which reacts from water from the soil. And when they react together, they produce glucose and also oxygen. And that is the reactants and products of photosynthesis. To react carbon dioxide and water requires energy, and this energy comes from light. The light energy is absorbed by a photosynthetic chemical called chlorophyll, which is found in chloroplasts. So photosynthesis occurs in chloroplasts that contain a green substance called chlorophyll, and that chlorophyll absorbs the energy transferred by light. And since the energy enters from the surrounding, the products of photosynthesis have more energy than the reactants, so this is a endothermic reaction. The reaction is known as photosynthesis because you are synthesizing glucose using energy from the light. So using the spec, the definition of photosynthesis in plants and algae is an endothermic reaction that uses light energy to react carbon dioxide and water to produce glucose and oxygen. And then that glucose that is produced in photosynthesis can be used to release energy in respiration by plants. So animals consume glucose to release energy, but plants have to produce their own glucose in photosynthesis. They could also use the glucose to make plant materials, which is their biomass. Why are leaves of water lilies broad and flat? Because they're broad and flat, they have a larger surface area, and that means they could absorb more light energy so that they could do more photosynthesis. So you need to be able to explain how the structure of a leaf is adapted for photosynthesis and gas exchange. So if you look at a cross section of a leaf, you'll notice that the cells are different to each other, so they have specialized functions. So first we've got to name the different structures of the leaf. So we've got the upper epidermis cells, palisade cells, spongy mesophyll cell, guard cells, and then we've got the gap in between the guard cells known as stomata. Then how are these cells adapted for photosynthesis and gas exchange? The upper epidermis is transparent, and this means that light can pass through it so that it can reach the photosynthetic cells. The palisade cells contain lots of chloroplasts, and this is because they're located near the top of the leaf, and so they absorb the most light energy for photosynthesis. These palisade cells are also regularly shaped, so they're very tightly packed to contain as many palisade cells together near the top of the leaf. The spongy mesophyll cells are irregular shape, and this creates air spaces, and that airspace increases the surface area so that it's easy for gases to diffuse into the photosynthetic cells. So an example is carbon dioxide diffusing into the cells so that they can do photosynthesis. The guard cells open and close the stomata, and this allows gas exchange to occur. So carbon dioxide could diffuse into the stomata, whereas oxygen and water vapor could diffuse out of the stomata. And that is gas exchange. So when are stomata more likely to be open? When the stomata open, they allow carbon dioxide to diffuse into the airspace so that they can diffuse into photosynthetic cells to do photosynthesis. The carbon dioxide has to react with water, and this requires light energy, so it doesn't make sense to open the stomata to diffuse carbon dioxide inside if there is no light energy. So the best time to open the stomata is when there is light. So how does the stomata open when there is light and close when it is dark? First, water moves into the guard cells, and it moves into the guard cells by osmosis. The guard cells then become turgid, in other words they swell up, and this changes their shape, causing the stomata to open. During dark, when there is no light, the opposite occurs. Here, water moves out of the guard cells by osmosis. The guard cells become flaccid, in other words they shrink, and this causes the stomata to close. And here's a summary of that information. Why do plants grow better in the summer? Well, clearly in the summer there is more light energy. Remember, light energy is required to synthesize glucose because it helps carbon dioxide and water react, because photosynthesis is an endothermic reaction. So light is one factor that could increase the rate of photosynthesis, but there are other factors such as carbon dioxide and temperature. So how do these factors affect the rate of photosynthesis? In your exam, you've got to be able to describe the relationship between those three factors and the rate of photosynthesis. You've got to also explain them, and then you also explain the effects of limiting factors in photosynthesis. So what's the limiting factor? First, we've got to think about what is the ideal combination of factors so that there is the maximum rate of photosynthesis. So if you give a plant enough carbon dioxide, enough light, and the optimum temperature for the enzymes in photosynthesis, then we'll notice that we get the optimum plant growth. So enough light, enough carbon dioxide, optimum temperature. And those are the combinations of factors for the maximum rate of photosynthesis. But what happens if we restrict one of these factors that affect the rate? So if we decrease the carbon dioxide concentration, you notice that the growth is not optimal. And it's the same thing if we decrease one of the other factors. We'll notice that we don't get optimal growth. But if we give the ideal factors, then we get the maximum rate of photosynthesis. So effectively, when we restrict one of these factors, we don't get the maximum rate of photosynthesis. This means it limits the rate of photosynthesis, so that factor is the limiting factor. So a limiting factor is a factor that limits the rate of photosynthesis. In other words, it limits the growth of photosynthetic organisms. So now, using that information, we should be able to describe and explain the effects of those three factors on the rate of photosynthesis. The brighter the light, the more light energy there is. So will photosynthesis be faster or slower? So here in this experiment, when you increase the light energy, you notice that the plant is producing more oxygen bubbles, which is collected by the syringe. And this is because the plant is doing more photosynthesis. So here, when we increase the light energy or the brightness, the rate of photosynthesis increases. So to explain that, when light intensity increases, the rate of photosynthesis also increases, and that is because light intensity is the limiting factor. It's the factor that is limiting the rate of photosynthesis. So when you increase it, the rate of photosynthesis increases. However, at a very high light intensity, the rate of photosynthesis stays the same, and that is because one of the other factors is a limiting factor. So that could be carbon dioxide, and or it could be temperature. So an increase in light intensity increases the rate of photosynthesis until a limiting factor stops further increases. The concentration of carbon dioxide in the air is approximately 0.04%. So why do commercial greenhouses have a higher concentration of carbon dioxide? So you can see, how does carbon dioxide affect the rate of photosynthesis in this graph? And then to describe and explain it, the more carbon dioxide there is, because carbon dioxide is a reactant, the more photosynthesis occurs. And then we've got temperature. Now we know that temperature affects enzymes, and enzymes are involved in photosynthesis. So when you increase the temperature up to the optimum, we know that the rate of reaction increases, and that's because there are more kinetic energy between the enzyme and substrate, so there's more successful collisions. Above the optimum temperature, an increase in temperature decreases the rate of reaction because the enzyme denatures. So we should be able to use this idea for how temperature affects photosynthesis. We can see we get a similar shaped graph. And now to explain this, when you increase the temperature up to the optimum, the rate of photosynthesis increases, and that is because temperature is a limiting factor. By increasing the temperature, there's more kinetic energy for enzymes and substrates involved in photosynthesis, more successful collisions, and therefore the rate of photosynthesis increases. Above the optimum temperature of the enzymes involved in photosynthesis, we know that the rate of photosynthesis will decrease, and that's because those enzymes denatures. And then if we go back to carbon dioxide, we know that when you increase the carbon dioxide concentration, the rate of photosynthesis increases, and that is because carbon dioxide is a limiting factor. It's the factor that is limiting the rate of photosynthesis. Very high carbon dioxide concentration has no effect on the rate of photosynthesis because there is another factor or factors that are limiting the rate of photosynthesis, most likely temperature, or there could also be light. So define limiting factor and explain what factors might be limiting at each stage. So this is an example of an exam question. We know limiting factors are factors that limit the rate of photosynthesis. So at A, you'll notice that there's 20 degrees Celsius. If you increase it 30 degrees Celsius, the rate of photosynthesis is higher. So therefore, the limiting factor at A must be temperature, because at high temperature, the rate of photosynthesis was higher. If you look at B, you could see that when you increase the light intensity, the rate of photosynthesis increases. So at stage B, light must be the limiting factor, because increasing light intensity also increases the rate of photosynthesis. And then at C, we'll notice that further increases of light intensity has no effect on the rate of photosynthesis. So it can't be light intensity that's the limiting factor at C. So it must be carbon dioxide. It could also be temperature, because we might have not also reached the optimum temperature. So a temperature above 30 might also increase the rate of photosynthesis, but most likely is carbon dioxide. And here's a summary of all three factors and how they affect photosynthesis. When light intensity increases, the rate of photosynthesis increases. That is because more light energy is absorbed to react carbon dioxide and water. And this is because light is a limiting factor, because it limits the rate of photosynthesis. When light intensity is very high, the rate of photosynthesis doesn't increase, because something else is a limiting factor, such as carbon dioxide and or temperature. When carbon dioxide concentration increases, the rate of photosynthesis also increases, and this is because carbon dioxide is a reactant. The carbon dioxide is the limiting factor, so it's limiting the rate of photosynthesis. When carbon dioxide concentration is very high, the rate of photosynthesis does not increase, because something else is a limiting factor, which could be light and or temperature. And then for temperature, when you increase the temperature, the rate of photosynthesis also increases, because there's more kinetic energy for successful collisions between enzyme and substrates in photosynthesis. Therefore, temperature is the limiting factor, because it limits the rate of photosynthesis. However, at a very high temperature above the optimum, the rate of photosynthesis decreases, because those enzymes involved in photosynthesis denature. Now when you see that the light intensity increases, the rate of photosynthesis also increases. So here, one variable increases, the other variable increases at the same rate. And this means that they are directly proportional. So while light intensity and rate of photosynthesis are directly proportional, we could say that if light intensity doubles, the rate of photosynthesis must also double. Now in this exam question here, you could see that when the distance from a light source increases, the rate of photosynthesis decreases. So here we're relating the distance from the light and the rate of photosynthesis. So in this example, an increase in one factor causes a decrease in the other factor. So they're not directly proportional, they are inversely proportional. So the distance from the lamp and light intensity are inversely proportional to each other. This means that when the distance increases, the light intensity decreases. However, light intensity decreases in proportion to the square of the distance, and this is known as the inverse square law. So we could say that light intensity is proportional to 1 over distance squared. So if you got a distance a pondweed is away from a lamp, and we were to half that distance, we could say that the light intensity now is 1 over half squared, which is times 4. So in other words, when you half the distance, the light intensity is 4 times greater. And we know that light intensity and the rate of photosynthesis are directly proportional to each other. So if the light intensity increases by a factor of 4, the rate of photosynthesis must also increase by a factor of 4. So if the rate of photosynthesis was 100 bubbles per minute, it will now be 400 bubbles per minute when you half the distance. In another example, if we were to double the distance, the light intensity now would be 1 over 2 squared, which is a quarter. So in other words, doubling the distance means that the light intensity is a quarter of what it was before. And since the light intensity and rate of photosynthesis are directly proportional to each other, the rate of photosynthesis is also a quarter of what it was before. So if we started off with 100 bubbles per minute and we doubled the distance from the light source, that means now it would be a quarter. So in other words, it would be 25 bubbles per minute. So just to summarize, when you double the distance, the light intensity is a quarter because it's 1 over 2 squared. If you were to triple the distance from the light source, it will now be 1 over 3 squared, which means it's 1/9 of light intensity. If you were to quadruple the distance from the light source, it would be 1 over 4 squared, so it would be 1/16 of the light energy. And we know that rate of photosynthesis is proportional to light intensity. So if we started off with 100 bubbles per minute, we know that doubling the distance means that you'd get a quarter of the rate of photosynthesis, so 25 bubbles per minute. If we triple the distance, it would be a third, so it's 11 bubbles per minute. If we quadruple, it would be 6 bubbles per minute. So you should be able to use calculations in the exam where you understand that light intensity and rate of photosynthesis are directly proportional to each other. However, the distance from the light is inversely proportional. Now, one of the investigations that you need to be able to explain is how light intensity affects the rate of photosynthesis. So here you're using algal balls that you place at different distances from the light source, and you investigate the effect of light intensity on the rate of photosynthesis. And that involves measuring the rate and when comparing the distance away from the light source. To understand this practical, you have to have an understanding of carbon dioxide and acid rain. So carbon dioxide reacts with rainwater, and that makes the rainwater acidic. And when it's acidic, it reduces the pH. So the more carbon dioxide that reacts with rainwater, the more acidic that rainwater is, and that means it has a lower pH. And then the opposite, if there is less carbon dioxide reacting with rainwater, it will be less acidic and a higher pH. So the more carbon dioxide there is, the more acidic it is, and if it's less carbon dioxide, it's less acidic. So we can use that idea when investigating the effect of light intensity on photosynthesis. So first you have a solution that you can use to measure the amount of carbon dioxide there is, and that indicator is hydrogen carbonate. You place algae balls in the hydrogen carbonate indicator solution, and then you have a light where the algae balls are at varying distances away from the light source. The solutions that are closer to the light source have a greater light intensity, and therefore they're doing more photosynthesis. And because they're doing more photosynthesis, they're removing the carbon dioxide from the hydrogen carbonate solution. So that means there's going to be less carbon dioxide. And if you decrease the carbon dioxide, that means that the solution has an increase in pH, which means that the solution becomes less acidic or more alkaline, which you can see from the change in color of the hydrogen carbonate indicator. This is the opposite for those hydrogen carbonate solutions that are further away from the light source. So here, there's an increase in carbon dioxide levels in the solution. And that's because more carbon dioxide has been added to the solution by the algae balls. So if they're adding more carbon dioxide, it's because they're respiring more then they are photosynthesizing. And this makes the solution have a decrease in pH. So in other words, the solution has become more acidic or less alkaline. And we can see that by the change in color of the hydrogen carbonate indicator. Now you could have a bottle wrapped in foil that doesn't absorb any light, and that could be used as a control experiment where you're expecting no change in color. And this proves that it's the light that's causing the change in the color of the hydrogen carbonate indicator, because the algae balls are using that light energy to do photosynthesis, where they're either removing carbon dioxide from the solution, or if they're doing more respiration, they're adding carbon dioxide to the solution. And that causes the change in color of the hydrogen carbonate indicator solution. So here the independent variable is light intensity, the dependent variable is the change in pH, which is measured using the hydrogen carbonate indicator solution. Any control variable could include things like temperature, or the volume of hydrogen carbonate indicator solution, or the number of algae balls that are used. As mentioned, the control experiment would be wrapping a solution in foil so it doesn't receive any light, so you're expecting no change in color. And then for the risk assessment, to make these algae balls requires the use of calcium chloride. That calcium chloride is an irritant, so therefore you don't want it to come in contact with your skin. So in the exam, if they ask for risk assessment, you should mention that you should wear gloves because the calcium chloride to make the algae balls is an irritant. That hydrogen carbonate indicator contains carbon dioxide dissolved in solution, so therefore carbon dioxide isn't a limiting factor. There's enough carbon dioxide. And the algae photosynthesize, removing the carbon dioxide, and that's detected by the change in color of the hydrogen indicator. And then you can see if it's become more acidic or less acidic due to the change in color. You could also do an investigation of the effect of temperature on the rate of photosynthesis using these algae balls, where you'd put the solutions in the container, and the container would be placed in water bath. And here the control variable would be light intensity. Other ways of measuring the effect of light intensity involves using a gas syringe to collect the oxygen bubbles, and that's because you could also count the number of oxygen bubbles, but measuring the volume of oxygen is more accurate, and that requires the use of a gas syringe. And then you could calculate the rate of photosynthesis, because it's the volume of oxygen produced in a certain amount of time, and that could be compared using varying light intensities. And this is everything you need to know about photosynthesis from the spec. The roots have two functions in plants. They could anchor a plant to make it more stable, but they also have root hair cells in the roots, and those root hair cells are adapted because one of the functions of the roots is to absorb water and mineral ions. If we look at a root hair cell, we could see that it's got a surface where water and mineral ions are absorbed through the membrane. And these root hair cells are adapted because they have this extension known as a root hair. And that root hair extension increases the surface area so that the root hair cells can absorb more water and mineral ions. So that is the adaptation of root hair cells. So how are the water and mineral ions absorbed by plants in those root hair cells? Well, the water moves from a higher water concentration in the soil to a lower water concentration in the root hair cells, and that occurs by osmosis, where water moves from a higher water concentration to a lower water concentration through a partially permeable membrane. You could also state that the water moves from a lower solute concentration to a higher solute concentration through a partially permeable membrane as well. So here's an exam question, just summarizing what I've just said. So that's water. So how does minerals get into plant root hair cells? In the soil, the mineral concentration is lower, and they need to move into the root hair cells where the mineral concentration is higher. So how could the minerals move from a lower concentration to a higher concentration? They use active transport. So minerals get into plant root hair cells by active transport against their concentration gradient. So how does the structure of a root hair cell, how are they adapted to support active transport mineral ions? Well, that's because root hair cells contain lots of mitochondria. Because they contain lots of mitochondria, that mitochondria can be used to release energy from respiration for that energy to be used in active transport. So here's a 6-mark question. Explain how active transport and diffusion provides a plant with the named substances it needs for growth. So you need to be able to explain that active transport requires energy to transport mineral ions such as nitrates from the soil into root hair cells from a lower concentration to a higher concentration against the concentration gradient. And then the other thing they also need is diffusion doesn't require energy because gases diffuse from a higher to lower concentration down the concentration gradient. The gas exchange happening in leaves occurs by diffusion, where carbon dioxide diffuses into air spaces into cells for photosynthesis, and then obviously oxygen could diffuse in for respiration. What is transpiration? Transpiration is the movement of water from the roots to the leaves, and then they come out of the stomata as water vapor. And to diffuse out as water vapor, that requires the water to evaporate. So effectively, transpiration is the movement of water from roots to leaves where it evaporates and diffuses out of stomata as water vapor. And the key word here in the exam is to use the word evaporate and diffuse when you're talking about the water vapor. You can see here the water molecules are absorbed in the roots, and then they go all the way up to the leaves and they evaporate and diffuse out of the stomata. So what are the factors that affect the rate of transpiration? What could increase or decrease how fast transpiration occurs in plants? So the 3 rates you need to know about from the spec are light intensity, air movement and temperature. You could get asked about humidity, which is realistically the opposite of air movement. So when are stomata more likely to be open? We know that when the stomata opens, carbon dioxide could diffuse into the air spaces such that they could diffuse into photosynthetic cells. They want to do that because it also requires light energy, because we don't want light energy to be a limiting factor. So there's no point opening the stomata for carbon dioxide to diffuse in unless there is light energy. So that happens when there is light. The problem is, if you open the stomata, water vapor will diffuse out. So therefore, when there is more light, the stomata will open, but this will also cause water vapor to diffuse out. So when you increase the light intensity, the rate of transpiration increases, and that is because more stomata will be open. So that's what needs to be stated in the exam question. How can you increase the rate of diffusion? Using the example shown. So here we could see that you could increase the concentration gradient, which is the difference between 2 concentrations. And when you do that, the rate of diffusion will be faster. The greater the difference between concentrations, the faster the rate of diffusion. And we could use this idea to explain how air movement affects transpiration. When it's more windy, there is more air movement, and that air movement blows away the water vapor outside the leaf. And because it blows away the water vapor outside the leaf, there is a bigger difference in concentration of the water vapor inside the leaf and outside the leaf. And because there's a bigger difference in concentration, that means that the concentration gradient has increased. So effectively, when you increase the air movement, the rate of transpiration increases because there is a bigger concentration gradient of water vapor. When you increase the temperature, this increases the kinetic energy of particles. That means the particles move more. We know that in transpiration, the water particles move from the roots to the leaves and then evaporate and diffuse out the stomata. So effectively, increasing the temperature increases the rate of transpiration because the water molecules have more kinetic energy and move faster when they diffuse out of the stomata. The key word here in the exam is to talk about the kinetic energy and the fact that the water molecules move faster when diffusing out of the stomata. So here is a summary of exam answers for all three factors. When light intensity or air movement or temperature increases, the rate of transpiration increases. In the case of light, it's because more stomata are open. For air movement, that air movement blows water vapor away from the stomata and that increases the concentration gradient. Whereas in temperature, the water molecules have more kinetic energy and move faster. And then in all three factors, this increases evaporation and diffusion of water vapor out of the stomata, which increases the rate of transpiration. Note that in the exam you want to talk about evaporation and diffusion of the water vapor out of the stomata, which gives you 2 marks, and then you get an additional mark for talking about the fact that the rate of transpiration increases. And then if you mention why, so more stomata open or increasing concentration gradient or more water molecules moving faster because they have more kinetic energy, you get an additional mark. The rate of transpiration can be measured using a potometer. And here you have a litre chute that is attached to the potometer, and then you change a factor such as light, air movement or temperature, and you can measure the transpiration rate by how fast the bubble moves. And that's because that's indicating how fast the water is moving into the plant to replace the water vapor that is diffusing out of the stomata. And you can measure how fast the air bubble moves by using the equation distance moved divided by the time it takes. So in an example here, a potometer was used to estimate the transpiration rate of a plant cutting. The bubble moved 25 millimeters in 10 minutes. Estimate the transpiration rate. So the distance was 25 millimeters and the time taken was 10 minutes. So it's 25 divided by 10, which equals 2.5. And then for the unit, because the distance was millimeters and the time was in minutes, so it's millimeters per minute. Now when the water goes through the leafy shoot, it then evaporates and diffuses out of the stomata, and that water is then replaced by the water in the potometer that causes the bubble to move towards the leafy shoot. So in the exam they might change conditions like having a greater amount of light intensity or more air movement or an increase in temperature, and that will cause the bubble to move faster or a greater distance in the same amount of time. And you should be able to explain that using the explanation in the summary. And we can see that here. So if we turn the light and fan off, you can still see that there is transpiration occurring, as shown by the bubbles, but it is very slow. I've speeded up this video. However, if we have both the light and fan on, the rate of transpiration should be a lot faster. So when I speed up the video, we could actually see that the bubble moves a lot faster. So that distance that it's traveling for the same amount of time is a lot more quicker. And that's because the air movement and light increases the rate of transpiration. So we know that transpiration is the movement of water from the roots to the leaves, where it evaporates and diffuses out the stomata as water vapor. So which specialized cells are adapted to transport the water and dissolved mineral ions as it moves through the transpiration stream? And you can link this to the experiment that was done on celery. So you could see that the dye moved all the way up to the leaves, and these specialized cells are known as xylem cells. So explain how the structure of the xylem cells are adapted to their function in the plant. So remember, their function is to transport water and minerals through the plant, through the transpiration stream that we saw from the roots all the way up to the leaves, where the water evaporates and diffuses out the stomata as water vapor. Now here's a xylem cell, and you can see the water and minerals are transported through the xylem cells, and that water collides with the inside walls of the xylem cells, applying pressure. Now the xylem cells are adapted because they have got fixed side walls to prevent the water pressure from having an effect on the side walls. They also have lignin, so both the fixed side walls that contain tough cellulose and the lignin, which makes the walls strong, prevent the xylems from bursting. The xylem are also cells that are dead, and because they're dead cells, they have no cytoplasm, and this makes it easier for the water to flow through the xylem. And that water flow is not slowed down between xylem cells because there is a lack of cell walls between one xylem cell and the next one. So those are the adaptations that you need to know about xylem cells: having a fixed side wall made of tough cellulose and strong lignin, preventing it from bursting due to the water pressure. Then the dead cells have no cytoplasm because that helps to allow water to flow through. And then there's a lack of cell walls between the xylem cells, so water flow is not slowed down. Now we said earlier that glucose can be joined together to make polymers such as starch, but they could also be converted to sucrose, which is transported around the plant. And that is because there are parts of the plant that doesn't do photosynthesis. So how would they increase their biomass if they don't have any sugars? So a good example of that is root. So some of the sugar that is made in the leaf has to be transported to other places such as the roots. So explain how the structures are adapted to their function in the plant to transport sucrose around the plant. Now this doesn't happen in xylem cells, it happens in phloem cells. So phloem cells are where sucrose are transported around the plant, and there are two different types of phloem cells. You've got the phloem companion cell and the phloem sieve cell. And when glucose is produced by photosynthesis, it can be stored as starch, and that glucose or the starch is then broken down or converted into sucrose that can be translocated, which means that it's transported to other parts of the plant. And when it's transported to other parts of the plant, that's known as translocation. And those phloem companion cells therefore need lots of mitochondria because they move the sucrose from a lower sucrose concentration to a higher sucrose concentration in the phloem sieve cell. So companion cells have got lots of mitochondria. That's one of their adaptation, because that mitochondria helps to release energy from respiration, so that active transport of the sucrose into and out of the sieve tube or sieve cells can happen. In other words, to move the sucrose from the phloem companion cells to the phloem sieve cells requires the use of active transport, because the sucrose has to move from a lower concentration to a higher concentration. Therefore, those sieve cells are adapted by having lots of mitochondria. And that means the sucrose can be pumped from the phloem companion cells to the phloem sieve cells. Now, to help that process, there are also pores between the phloem companion cells and the phloem sieve cells, so that the sucrose can move through. And then, due to the pressure of pumping the sucrose into the phloem sieve cells, the sucrose can either move up or down the sieve tubes. And there is more room for that to occur because those phloem sieve cells have very small amount of cytoplasm and they have no nucleus, and that creates space for the sucrose to move through the phloem sieve cells. Also, in between two phloem sieve cells, you have got holes or pores between them in the cell walls, and that makes it easier for the sucrose to translocate from one sieve cell to the next. So here's a 6-mark question. Explain how a potato plant transports materials so that starch is removed from the leaves and builds up in the storage organs. This is an example from the spec where you've got to explain how translocation of sucrose occurs. So first, as we mentioned, the starch is broken down to make, or is converted to sucrose. Then the phloem companion cells pump the sucrose into the phloem sieve cells by active transport, and that means that they need to have lots of mitochondria, which releases the energy for active transport to occur. There's also pores between the phloem companion cells and the sieve cells. Then pumping of sucrose causes a pressure for the content in the phloem sieve cells to either move up or down. Now those phloem sieve cells are adapted because they have pores between the cells, and therefore sucrose can move from one phloem sieve cell to the next. They also are adapted because they have no nucleus and only a small amount of cytoplasm, and that makes it easier for the sucrose to flow through. And then once it reaches storage organs such as potatoes, the sucrose is pumped out of the phloem sieve cells into the phloem companion cells by active transport and used to make starch. And here's a very big keyword that you need to use in the exam. That transport of sucrose around the plant is known as translocation. So there are two key words here, translocation and transpiration. In transpiration, water and mineral ions are transported around the plant. In translocation is sucrose, although that could include other sugars or foods. Transpiration happens through xylem cells, whereas translocation happens in phloem cells, which are the phloem companion cells and phloem sieve cells. In transpiration, the xylem cells are dead, but in translocation, these phloem cells are living. Transpiration occurs from the roots all the way up to the leaves, where the water evaporates and diffuses out the stomata, so the direction is upwards, whereas in translocation, the sucrose could either move up or down. And then transpiration involves the water molecules moving, whereas in translocation, to move the sucrose requires active transport because they move from a lower concentration to a higher concentration. So using those three cells and the other cells that you've learnt previously, you've got to know some functions and adaptations of the functions of all of those cells. So this is a summary that you could read for all of these. And then here's some additional summaries for the adaptation of cells in photosynthesis. So in this unit, there are lots of different types of cells you need to know about, and you got to know how they're adapted for both, or for photosynthesis, and also transpiration stroke translocation. Plant adaptations. So explain how plants are adapted to survive in extreme environments. So this could be examples like cacti that need adaptations to live in hot, dry conditions. And there are different types of adaptations you know about, such as the leaf size and shape and the stomata. But one of the adaptations you need to know about is the cuticle. And that waxy cuticle is thick in certain plants. And the reason why it's thick is because it reduces the amount of water loss by evaporation. So in other words, it means that there's less transpiration occurring because they have thick waxy cuticle. It reduces the water loss on the upper epidermis. It prevents water from evaporating out. Other adaptations include the leaf size and shape. So here you could have thinner or smaller leaves, or some plants have no leaves, instead they have spines. And in all of those examples, that reduces the surface area. So having thinner or smaller leaves or spine means that the surface area is reduced, and that reduces water loss by evaporation. So again, these are conditions where plants are adapted to survive in environments where there's not a lot of water. Now here's some more examples of differences in leaf size and shapes, and also including stomata as well. So in this case here, you could see that the leaf is either curled or rolled. Also, this leaf has got hairs. And then if you look at the example of the stomata, these stomata are sunken in pits. And in all of those examples, it reduces the air movement around the stomata. So in these locations. Now we know previously that air movement has an effect on transpiration, because one way of increasing the rate of diffusion is by increasing the concentration gradient. So if there's a great amount of air movement, that means that air movement is blowing away the water vapor outside the leaf. It's increasing the difference in concentration of water vapor inside the leaf and outside, and that increases the concentration gradient. So more air movement causes an increase in concentration gradient, which causes an increase in transpiration. So if you could reduce the air movement around the stomata by either having curled rolled leaves or hairs or stomata sunken in pits, effectively you're reducing the concentration gradient of water vapor, and this means that you're reducing the diffusion of water vapor out of the stomata. In other words, you're reducing transpiration. So these plants that have these adaptations could therefore live in areas where there's not a lot of water available. Some more adaptations, and this examples involve stomata. So some plants have got fewer stomata, and then some plants only open the stomata at night time, because at night time, because the temperature is lower and there's less light intensity, that means that the rate of transpiration will be lower. So that means it makes sense to open the stomata at night time to diffuse in carbon dioxide, and then that carbon dioxide is used at daytime for photosynthesis. But the stomata could be closed because they store the carbon dioxide. And both these examples, having fewer stomata or having stomata only open at night, reduces water loss by evaporation. So that's another example that some plants have to reduce transpiration so they can survive in areas that have less water available. And then another example of adaptation, this time involving roots, is having roots that are widespreading or deep. And when you have these adaptations, this means that you could reach water that is lying deeper, and those are the adaptations you know for plants in extreme environments. Now moving on to the last topic, plant hormones and their uses. So we know a stimulus is a change in the environment that you could detect. So we've got stimulus that humans and other animals can detect, such as a change in light or change in sound. But plants also have two stimulus that you need to know about for your exam. They could detect and change or react to light and also gravity. And these responses have got names. So a response to light is known as phototropism. A response to gravity is known as gravitropism. Now we learned previously that when plants grow, their cells get longer, and that is known as cell elongation, and that is a very key word when explaining the effect of plant hormones. So how do hormones respond to light? So if there's light all around, we could see here that you have got hormones on the tips of the shoots, and these hormones are called auxin, and they respond to the light. And when they respond to the light, where the light is all round, they diffuse equally to both sides of the shoot, and because they diffuse equally, both sides of the shoots have got auxin, and therefore that causes both sides of the shoot, the cells in those locations, to elongate, and when it elongates, the shoot grows towards the light, so it could do more photosynthesis. Now what happens if light is coming from one side? So here we've got light from the right side. Again, you've got auxins on the tips of the shoots, and they respond to the light differently in this occasion. Instead, more of the auxin diffuses to the shaded side, or away from the light. And when it diffuses to those cells, it causes those cells to elongate, so the cells on the shaded side will elongate more, and when they elongate more, that means that the shoot will bend and grow towards the light. So to summarize that, you can see that first you've got light from a certain direction, you've got auxins, and they're the plant hormones, and they're produced on the tips of shoots. They could also be produced in the tips of roots as well, and they respond to the light by diffusing unequally more to the shaded side, which is the side opposite the light. And when they diffuse unequally more to the shaded side, that means those cells will elongate more, so that side of the plant or the shoot will elongate more, causing the plant shoot to bend and grow towards the light, and this means that the plant can do more photosynthesis. So here's an example of how plants respond to light. They're not moving, instead they are growing towards a direction that helps them to do more photosynthesis. So in your exam, there are classic experiments involving auxin that you might have to describe and explain the results of. So we already talked about A, where you have a shoot and you have a light coming from a certain direction. The shoot bends towards the light. We know that because the auxins on the tips of the shoot diffuse unequally more to the shaded side, causing cell elongation, and that means the shoot bends and grows towards the light. In B, the tips of the shoot is covered with black paper. The result is there's no bending of the shoot. So this is because light can't pass through the black paper, and therefore the auxins can't respond to the light. So instead, when they diffuse, they will diffuse equally, causing cell elongation on both sides of the shoot. So that means that they don't respond to the light by diffusing unequally. They'll just diffuse how they normally would do on both sides of the shoot. So cells on both sides of the shoots will then elongate, so the shoot grows evenly on both sides, so it will grow upwards. So in this occasion, it's not growing, or it's not bending and growing towards the light. Instead, it just grows upwards. In C, you can have the tip of the shoot covered with transparent cap. Now because it's transparent, that means that the end result of the shoot bends towards the light. This is because it's transparent, the light can pass through, and therefore the auxins would diffuse unequally more to the shaded side, causing those cells to elongate. That means the shoot bends and grows towards the light. In experiment D, the tip is removed. Now we know that the tip contains auxin, so if you remove the auxin, there is no auxin to diffuse to towards the below towards the shoots. So that means that there's no response. So there's no bending. So in this example, the shoot tip where auxins are produced are removed. Therefore, there's no auxin to elongate the cells, and that means that the shoot doesn't grow. And that is proof that the auxin is produced in the tip. This also proves that the auxin is the chemical that causes the cell to elongate. In experiment E, the tip is removed, but this time it's put back, but it's put back on an impermeable block. So impermeable means that it can't diffuse through it. So the end result of the experiment is that there's no bending of the shoot. So the impermeable block prevents the auxins diffusing to the cells, and therefore, because these cells don't have any auxin, they will not elongate. And then experiment F is similar to E, but this time when you replace the tip back, you're putting on an agar block, and agar allows auxins to diffuse through, so the end result is the shoot bends towards the light. So here, auxins can pass through the agar block because that agar block is permeable, so it diffuses unequally more to the shaded side because it's responding to the light that's coming from the right, and that means those cells on the shaded side will elongate more, and that causes the shoot to bend and grow towards the light. So those are classic experiments that you might have to describe and explain in your exams. Now there are further key words you want to use when explaining the reaction of the plant shoots, but also the roots. So shoots grow towards the light. We call that positively phototrophic, and that is because those shoots are responding towards the light. It's positive and it's in the same direction. They're growing in the same direction as light, so it's phototrophic, so it's positively phototropic. Have the roots grow away from the light. So because they're growing away from the light, their response is known as negatively phototrophic. So it's negative because it's away from the light, and it's phototropic because it's a response to light. Now gravity also has an effect on how plants grow, and this is known as gravitropism. So we said that auxins are also found on the tips of roots, not just on the shoots, and they have the opposite effect on roots. So they do the opposite effect in roots. In other words, instead of causing cells to elongate, they stop cells from elongating. And this means that this causes positive gravitropism. A positive gravitropism is when the growth is towards the direction of gravity. It's positive because the growth is in the same direction as it is to gravity, and it's a response to gravity, so it is positive gravitropism. So the key word here in an exam question about gravity is the fact that auxins, which are found in the root tips, instead of causing cell elongation, they inhibit cell elongation in the roots. So their effect in the roots is opposite to their effects in the shoots. In the shoots, they cause cell elongation. In the roots, they inhibit cell elongation. So how does gravity have an effect? So here, the auxins move from the root tips to the underside of the plant root, and they inhibit the elongation of root cells on the underside. However, the upper side root cells will elongate, and that means that the roots will grow downwards towards gravity, which is known as positive geotrophism or positive gravitropism. Just a reminder that the effect of auxin is the opposite in the roots. So in the roots they inhibit cell elongation, however in the shoot they increase cell elongation. So here's an animation to explain that. How do plants respond to gravity? So you can see here there is auxins on the tips of the shoots and roots, and because of gravity, the gravity makes the auxins stay on the underside or the lower side of both the shoots and roots. However, on the shoots, they cause the cell to elongate, so these cells will become longer, and then when it bends, it is growing in the direction of light, but it's going in the direction away from gravity, so it is positively phototrophic, however it's negatively geotrophic. And then if you look at the roots, you can see the roots, they cause the auxins are on the lower side or the underside of the cells. they inhibit cell elongation. That means there is more cell elongation on the upper side of the roots, and that causes the roots to bend and grow downwards, and because it's growing downwards, you can see it's positively geotrophic. So shoots grow away from gravity. You can see that means they are negatively gravitrophic or negatively geotrophic, and then roots grow towards gravity. They are positively gravitrophic. And then here is a summary explaining all those responses. So you can see that the light has an effect on the shoot. The auxins diffuse away from light, causing cells in the shaded side of the shoot to become more elongated, and that results in the shoot bending towards light, and it becomes positively phototrophic. You can read the summaries for the other 3 examples. And there's further summaries here as well. The last topic is uses of plant hormones. So how can you make green unripe bananas ripen faster? You could do this by placing them next to ripe bananas, and that's because ripe bananas can release a hormone called ephine that helps fruit to ripen. So in the spec there are 3 different plant hormones that you need to know the uses of. They're called auxin, gibberellins and ephine. 2 uses of auxins that you need to know about are selective weed killers and rooting powders. In selective weed killers, the auxin kills only broadleaf plants, which are weeds, but it does not kill useful narrowleaf crops such as wheat. And this is because the broadleaf plants absorb more auxin, causing them to grow uncontrollably, and that means that they die. In rooting powders, the plant cuttings dipped in rooting powder speeds up the growth of roots. And that's useful because if it speeds up the growth of roots, you could use that in asexual reproduction, which is quicker instead of growing using seeds. And that's why rooting powders are used in tissue culture, which we learned in previous units. The next hormone is gibberellins, and there are 4 different uses that you need to know based on the spec: seed germination, fruit formation, flower formation, and seedless fruits. In seed germination, you can use gibberellins to start germination of seeds to help it grow roots and shoots without there being a period of either darkness or dryness. In fruit formation, gibberellins helps to produce bigger fruits. For flower formation, plants could flower without photoperiodism. In other words, plants usually flower because they respond to the number of daylight hours in day, but you could do this by using gibberellins instead of having plants flower naturally. And you can also use gibberellins in seedless fruits because the fruits will grow without seeds inside. And then the last hormone which we talked about was ephine, which is also known as ethylene in some books. And their use of ephine is to ripen fruit. So here it helps fruit to ripen, and that's useful. Is in the exam you might have to mention that the unripe fruit can be transported. It's less likely to damage or get bruised. And then using ephine you can ripen it later when it's required by the consumers without it rotting.